here we are. Okay, so we're there. Right, so I call the picture I use the same. It's a wonderful photo or a series of a collage, and it's really about what cells are, more a little bit on how they operate, more on genetics, and some stuff on heredity. So I'll call it part 2, and it just expands what we did last time, more detail, and I'd like to go into the 3 types of organisms. So we've got the prokaryotes, they're the ones without a nucleus, the eukaryotes, which do have nucleus, but they have a cell wall, and animals like us, where we are have everything inside ourselves, just like plants. Okay, so the standard start is all living things are made up of at least one cell, anything less than one cell, you're either a prion or a virus, or a couple of other things as well. But, okay, so viruses are non-living. They get all the energy for reproduction from wherever they attack, whatever they attack, and they're also they depend on their hosts for being moved around, as we know with COVID. So what we'll look at different types of organisms, and we'll have a look at the similarities and differences. So let's have a look at I'm leaving viruses out of this completely. So bacteria, we call them prokaryotes, the first cells. They don't contain any internal structures whatsoever. They have cell walls, different to plant cell walls, but they're full of the enzymes that they need to use, and they can organize their own enzymatic reactions. So they can do everything that we can do, but in a slightly different way. So they metabolize, they reproduce. Okay, so they don't have nuclei, they do not have organelles like we do, and they don't have a cytoplasm. They've got what's called the protoplasm. That means the first plasm, the fluid that's inside cells. Now, they share certain features with us. Sorry, no, that's too soon. They share common features between all of them. One, they lack a nucleus, and they don't have any membrane-bound organelles, yet they do everything that we do. They have one circular DNA, and it's naked, it's not bound by proteins like our DNA. So it's circulated, having no structure, whereas ours does. And they have some plasmids, which are little bits of DNA, and this is how they communicate with each other and pass on information to each other. And it's the plasmids that we use to be able to produce molecules for medical purposes, like insulin. All insulin now uses plasmids or uses bacteria. Now, they have small ribosomes, and it's the ribosomes which are involved in protein synthesis. So they have that, well, they couldn't produce proteins otherwise. Now, there are different types of bacteria, and they have certain characteristics. So their shape, for example, some are round, and they're called cocci, or coccus is singular. So they're rod-like, the little sticks, they're called a bacillus. They look like little commas, some of them, they're the Vibrio, and some of them are spirals, the spirochetes or the spirula. The cell wall is gram. Now, there are two types: the gram positive and the gram negative. Now, the peptidoglycans is a mixture of, or the combination of protein and sugar, whereas that's for the gram negative. So the gram positive, the gram negative is a mixture of fats and polysaccharide, which is a different sort of sugar. So these are protein and sugar, and this is fat and sugar. And that's why with gram, they have, in order to be able to treat you, and the doctor may not be exactly sure what's going on or what sort of bacteria you've got, you get sent away for testing. And virtually all antibacterial medicine relies on the disruption of the cell wall. So they have to know whether it's a gram positive or gram negative. And it's called that because of the way they react to a stain called the gram stain. I think that was named after somebody. And then they have different gaseous requirements. So there are an anaerobic bacteria and aerobic bacteria. And the anaerobes are either obligate, which means they have to have no oxygen, or they can survive, but they're just as happy without it. And the nutritional patterns are, some of them are autotrophs, which means that they can photosynthesize or they can chemosynthesize. So they use something like light or some chemical to produce the energy that they require, or the heterotrophs, which means they have different mechanisms of obtaining their food. So they look different. That's the spheres here, the cocci, the circular. They don't usually occur singly. They often occur as doubles, the diplococci. So you may have heard of the streptococcus, the pneumonia, and they're the short ones. The longer strips are like a little string of balls. You can have staphylococci, they're like bunches of grapes. Staphylococcus aureus, that's golden staph. That's a really nasty one, easy to pick up in hospitals, and often a killer if it's not picked up early enough. And then you've got bacillus in chains. That's the anthrax. You have some with flagella, and they can move around. The salmonella, the typhoid is one of them. It's a typhoid. Some of them produce spores like Clostridium botulinum, and that's you end up with botulism, and that's a killer. That's the one. It's an anaerobe. It'll live perfectly happily in, say, a can of something, or something that's produced at home, which hasn't been cooked properly. So some may still be there. They will survive without any oxygen, and when they run out of food, they will just produce little spores. But the botulism is a chemical that's in there. So if you ever buy a tin from somewhere, a can, and you notice that the top, instead of being flat, a little bumpy, do not under any circumstances open it and send it up to the health department. That could well have a botulism in it, and they need to be able to then trace where the other cans from that batch have gone to. Really nasty. Then you've got the Vibrios, which can swim around. These are the spirals like that, and Helicobacter pylori, which is the bacterium that have been discovered to cause stomach ulcers. And spirochetes, they're like little worms that just move around. The cholera, this is Vibrio, yeah. So inside a bacterium, well, from the outside in, you've got layers of walls. There's a capsule, it's usually some sort of a polysaccharide, sugar. You've got a cell wall, which is a different sort of material. And then you've got a membrane inside that. You've got, now notice there's no nucleus. There's one plasmid drawn in here, but you usually have a large number. The DNA, the chromosome is fairly large, but it's not tightly wound up like ours. It can have a flagellum, which helps it swim. It had pili, a singular is pilus. So it can also move around where they all move in the same direction, and also senses things around it. So it's fairly simple, but you know what? It does everything we do. When they reproduce, they just split into two. It's called binary fission. They reproduce everything that's in them, inside them, and then they just membrane and walls and everything. It's formed in the middle, and they separate, and then you've got two. So, and they're called daughter cells, of course. That's in all forms of cell reproduction. The binary fission that here starts off with the DNA replicates. The bacterial cell elongates and splits into two, and then they have, of course, all the identical DNA to the parent cells, but also the plasmids, which I should have mentioned here. They reproduce as well very quickly and split and move around. And they're a slightly different sorts of plasmids, but I won't worry about that. Now, each daughter cell is a clone of the parent cell, identical. So here's basically your original bacterium, and it starts reproducing the DNA, and it just goes into two. While it's doing that, the cell extends itself, and you end up with the two cells. They haven't separated yet, but it's got the DNA there, and then you've got two. But it leaves out a lot of other things. And by the way, here it says a septum forms. Now, septum is a divider. So, for example, if you think about your nose, you have two nostrils, and they're separated by a septum, just a standard word, and you end up with two. Now, under ideal conditions, like the right temperature, the right nutrients, and so on, Escherichia coli, E. coli, that's one of the big ones that are in our gut. It can divide every 20 minutes. So this process takes about 20 minutes with E. coli. After about 8 hours, if it just continuously reproduces, you end up with over 16, close to 17 million cells. And that's reproduction. And so if we are, depends on the bacteria, they're all a little bit different, but it's within that framework. And these pathogenic bacteria can reproduce very, very quickly in us and cause quite horrendous situations. Now, some bacteria produce spores, and these spores allow it to become dormant when situations are not quite right for it to just survive. So by forming a spore, then they hibernate. They have minimal metabolism, and and very minimal respiration, and everything's reduced. So they exist, but just it's almost like if you think of an animal that hibernates, or a tree that a plant that has lost all its leaves over winter. And I'm not sure if you know, by the way, why some plants are deciduous and some are not. The ones that are deciduous, as in the same situation as the bacteria, conditions are not conducive for them to live in. So they mostly, I think they all come from areas where there's no liquid water. So in countries where in winter the water in the soil freezes, the plant can't access it. So by dropping all its leaves, it goes into a level of dormancy. It's still alive, it still functions, but very minimally. And and then once the water in the soil starts to melt, and it gets a bit warmer and a bit more sunlight, it comes out through there, and the leaves start to regenerate. The same with the bacteria. Okay, so the spore formation is the same idea as dormancy in with animals. I don't know, like bears, for example. They all the metabolism and everything is reduced. Now, so this sentence is basically what I'm saying. So by going into this spore formation, it allows it to survive through long periods of, we call it environmental stress. Basically, there's not enough material, therefore, for them to survive on. But the endospore itself, the spore around the bacterium, is very tough, and it just can hang around for many years. And so the Clostridium botulinum is one of those. Now, so it contains the bacteria's DNA, the ribosomes, and all the chemicals which will keep it in dormancy. Dormancy means still alive, of course, but so it's pretty well organized. So here's an idea of so the core here, as you see in this, it's got its DNA, it's got its ribosomes, which it needs to produce proteins with, and it's got glycolytic enzymes, the enzymes that break down basically sugars, and it can survive for many years. The spore, it's got a membrane, it's got the wall of the spore, then it's a peptidoglycan, so it's a sugar with protein mixture, and then you've got another layer which is cross-linked. It's a totally different, the same material but a different structure, and then to the outside layer is a keratin layer, the protein. Now keratin, we've got a lot of it on us, for example, our hair, our fingernails, the skin, our skin cells have got a layer of keratin to protect ourselves. Now, bacteria communicate with each other beautifully. They have these little bits of DNA called plasmids, which I mentioned, and they replicate independently from the main circular DNA of the bacterium. And they are found mainly in bacteria, but they're also, they're mainly there, but archaea have them. And the archaea is a group of bacteria which are the next level in evolution after the bacteria that we're talking about now. There are modern bacteria. And then we've also got the eukaryotes like us. but the yeast and plants also have these plasmids. It's like tiny little circles of rings, if you like, of DNA. And these plasmids, they have a number of functions, and one of them is resistance to antibiotics. And the resistance itself may be already in a bacterium in the plasmid, or when there's a little bit of some antibiotic there, it may, when it produces the plasmids, it could mutate accidentally and produce a resistance. Now, once there is resistance, the bacterium will cause those plasmids to, with that antibiotic resistance in the DNA, to replicate and share them in their population. So they're pushed out of the cells and they're picked up by other bacteria. They also they degrade, or degradative functions, where they break whatever they're in down. They share that capacity, and also the virulence of them attacking whatever they're attacking, whatever cells, be it plant or animal, whatever it might be. As I said, they constantly share their plasmids, and that is how they communicate. Let's move now away from the prokaryotes and got the eukaryotes. Now they're the cells, the bacteria, the archaea, the bacteria. Besides them, the name eukaryotes are well and good, and from karyot, I'm not a Greek speaker. A nut or kernel, which means it's got a nucleus. So they're far more complex in structure than bacterial cells, and eukaryotes have, this is important, membrane-bound nucleus. Generally just one, but some of our cells, white blood cells, have multiple nuclei, as well as membrane-bound organelles. And this is what allows us to function in particular ways. These membranes are very, very important. So what sort of organelles do we have? And again, not in bacteria. So we have a nucleus, generally. It may contain DNA, so it does contain DNA and contains RNA. More details on all these as we go, but so the endoplasmic reticulum, very important. It's involved in protein production. The Golgi bodies, where they have protein transport. The mitochondria, where ATP, the energy is produced. Lysosomes, they are little bubbles, if you like, inside the cell, and they degrade things within the cell that are no longer required. There are also chloroplasts in plants where photosynthesis occurs. So here I found these two really pictures, this one and the next one. Now you can always tell an animal cell from a plant cell, because a plant cell has got a very distinct, different shapes, but they are a very solid shape, whereas animal cells are quite fluid. Now, if you go from the center out, where you've got the nucleus and you've got a nucleolus, and that is where the RNA is sort of basically stored when required. There are nuclear pores where the messenger RNA comes out, to the endoplasmic reticulum. This, the blue things here, and that's where our proteins are produced on that, and you can see little dots, and that is where on the dots, which are called ribosomal RNA. I'm not going to go into too much detail. Well, not yet. That's where the proteins are produced. The proteins are put together in the Golgi apparatus. Then they break off these little vesicles, and they're either used within the cell or secreted. So you can have secretions of enzymes, you can have secretions of hormones, or whatever it might be. These things, like sausages, notice they have double membranes, and within the middle internal membrane, this is where our energy is produced. You might notice, you may have noticed that the thing here is smooth in endoplasmic reticulum as opposed to rough. And when I first saw them under the microscope, the electron microscope, they saw these little bubbles, little points. So they call them rough. These ones didn't. The rough ones are where proteins are produced, and the smooth is where fats are produced. What else should I tell you? The centrioles, these things here, are involved in the replication. When a cell undergoes mitosis or meiosis, which we'll talk about later, these are important bits. The rest we can do without. Oh, you can see here, just so these things, which a thing called exocytosis, where the vesicles come to the membrane, they become part of this. It's exactly the same membrane around these as a cell membrane, and they become part of the membrane, and whatever they have inside them is released, and then it gets picked up. Now, the plant cells have much the same internal structure. So your nucleolus, nucleus, the endoplasmic reticulum, the Golgi, the mitochondria, but what they do have that animals don't is the chloroplast. And this is the bit that the organelle that undergoes photosynthesis. Now, not all plant cells have chloroplasts, because not all plant cells can photosynthesize. But the ones that do have them. So the cell membrane itself is a phospholipid bilayer. Now, bilayer means it's got two layers of this fat, which has a phosphate attached. I'll explain in a moment. And so these are done in such a way that molecules are transported in or out of the cell, and also vital for cell recognition. And we've talked about immunity, the immune system. This is, we talk about cell recognition. So the cell membrane is a barrier and keeps things in, keeps things out. So the cell can select what it releases and what it allows in, depending on what stage or whatever process it's undergoing. And it's about transport into the cell of nutrients and movement away, and movement, of course, but also wastes are removed. So it's very, very complex, and I think it'd be safer for me not to go into much detail there. But the basics are this. So you have a bilipid phospho layer. So there are two layers, one layer, two layers. There are two strands of, if you like, on the end one of fats here, and that's a phosphate molecule. And those of you who've done some chemistry, it's PO4 3-. And and that's called a phospholipid bilayer. And all our membranes are made of this. And outside the cell, it's called extracellular space and intracellular inside. Okay, now it's important to realize, and I just said that the head of these, if you like, the phosphate, has got a negative charge, which means it's hydrophilic. It loves water, whereas the fat has no charge, so it's hydrophobic. Now, so hydrophobic means it hates water. Hydrophilic, it loves water. And that's one of the ways we operate, by having these differences with every single cell. Now here's a slightly different version of the diagram. So you've got your phosphate, you've got your two fat layers, and then here you've got cholesterol molecules in between them. And these fat molecules, they're different to these fat molecules, give us pliability, give us mobility of the skin, and also is involved in gives us ability so the cells are not rigid. That's probably the best way to do it. So as opposed to the plant cells you mentioned, our cells are very fluid. Now, the phospholipid molecules, so the fat, the phosphate, you mentioned that, the fat is hydrophobic, it repels water, and the phosphate has a charge and attracts water. It's hydrophilic. I don't know why I left that word out. Now, they're very small molecules like water and the gases, they just fly through. There's no issue, nothing stops them. But larger molecules are affected, and like proteins. So they are stopped from crossing the membrane unless there's a specific section within a particular cell's wall or membrane, cell wall, cell membrane, I should say, where some specific molecules are allowed to be transported through. So, for example, insulin in the membrane is required for glucose to get into cells. So glucose can only enter a cell when insulin is it's embedded into the cell membrane. Some large molecules which are fat soluble can move through easily, but things which are proteins like insulin, they're not. There's a difference there. We also have the molecules which we call self. We talked about this before, and the cell membranes, they have self molecules which the immune system recognizes, which means that they get looked after, they don't get destroyed. If of course, if there are cells within our system which don't have the self molecules, then they are destroyed, they are removed. So this is a slightly different structure, a diagram of the structure. So outside and inside the cells. So inside is your cytoplasm, you've got your bilipid layer here. Then you've got these transport systems. This one depends what it is. This is basically what insulin looks like. It's like a tunnel, and glucose is transported through. The immune system ones are there, and every one of us has a slightly different molecule there. Every person, every animal, and this, so it shows you the variety. it's the end part that's the recognition system. Very, very complex. Let's have a look at the nucleus now. Now, the nucleus is a pretty complex organelle, and it contains all the information that needs to make us up, or any organism, eukaryote that is. It's got internally is where the DNA is kept and maintained. It's got the nucleolus, which is what I mentioned before, where the RNA is maintained. Now, there's a nuclear membrane, a nuclear envelope around it, and it's full of holes. And these holes allow, and I'll get to that a bit later, what I think they call messenger RNA, which is produced in here from genes, specific genes that come out, and they go onto the pores of the rough endoplasmic reticulum. And this is a beautiful diagram, really. It shows the relationship between the rough endoplasmic reticulum, the rough ER, and the nucleus and the nuclear pores. So the production of proteins can happen terribly, terribly quickly. Now, the information in the chromosomes within the genes, the information is in the form of a series of codes. And these codes are part of the DNA. There are other sections as well. So what have we got? And a little bit more detail here. So with the rough endoplasmic reticulum, these are the ribosomes. And there are 3 types of ribosomes. Now, you need, this is all for protein production. The endoplasmic reticulum it's quite flat. Here's your, if you go back, here's your endoplasmic reticulum, this orangey thing. I think they just slightly different diagram. So here are the pores, and I should mention, I did before, there's smooth endoplasmic reticulum which don't have the ribosomes. So they produce lipids and the steroid hormones. That's the steroids, basically fatty hormones. And they also are involved in the detoxification of harmful metabolic byproducts which we produce. So that's done right through the whole thing. So those 2 are there. There's another thing called the Golgi apparatus. Now these, the old name Golgi bodies, and they gather the molecules of the same sort, and they either combine them with others, make complex molecules, and they also finish off molecules. So they can have a fat added to the protein, they can have a carbohydrate added to the protein. So you have a lipoprotein with a lipofat protein or glycoprotein. And there are many, many variations of that. And then these become bigger and bigger molecules, and they're packaged in these tiny little bubbles called vesicles. And then they are either stored or they send them out of the body. And that's called exocytosis. I mentioned before, exo, external, cytosol cell. There's another version of that called the endocytosis, where the opposite happens with things outside the cell. The cell forms a little bubble and then seals it off, and it moves around the cell and goes to wherever it needs to go with whatever it's just been brought in. So this is showing the relationship that's sort of a general diagram of the Golgi apparatus. And this is a more inclusive sort of diagram. I don't think I need to mention any more here. Now, mitochondria and a singular mitochondrion, these are membrane-bound organelles. All right, so they are made of membranes, and they generate virtually all, probably 99.5% of our chemical energy, which we need for all our reactions. And this energy molecule is called ATP, adenosine triphosphate. And the mitochondria have their own DNA. And they can also modify their numbers according to need. Now, they have their own DNA. The idea goes that they were once microorganisms, like bacteria, long time ago, and they were picked up by new eukaryotic cells, ones with nucleus, and they're picked up and they form a symbiotic relationship, so that these little bacterial type things, which we call mitochondria, were protected, were fed, and then they were able to produce energy, which the protective or the protected cell, but no, the bigger cell, the eukaryote, was able to use this energy. The same thing is with chloroplasts. Their organelle that produces undergoes photosynthesis to produce sugars. They also have their own DNA. So here's a mitochondrion diagram of that is. It's got two membranes, double membrane, and it's got its own DNA, and so it's got its own ribosomes. It's got a nice little system, and if you notice the way these diagrams are drawn, they're little brown dots here are very close to each other. So when it undergoes metabolic process in the production of energy, it's the molecules that are being produced are just passed on from one to the other very quickly, like a conveyor belt, rather than just letting them loosely float around inside. They're very highly organized, and so we can produce energy quickly. So bacteria don't have them. They produce the energy in a slightly different way. So there are lots of enzymes in the internal membrane, like I just showed you, and they're in a very specific order, and they convert the various molecules into ATP. Now, ATP, adenosine triphosphate. So they have 3 phosphates, and the adenosine is a version of adenine, which is one of the nucleotides that make up DNA. So not adenine, adenosines. Okay. So adenosine triphosphate, and supplies energy for nearly everything that we do, everything. So here's the diagram, and this is quite important. So you've got the adenine or the adenosine here. So it's a nitrogen-based molecule. There's the sugar, and there are the 3 phosphates. And it's important to notice they've got 3 negative symbols there, and this yellow splotch indicates that that's the high energy bond. So when we're doing things like we stand up and walk, or if you want to go, even walking, but if you want to go for a run, or you pick up a heavy weight and you're just moving something from point A to point B, whatever we do requires, our muscles require this energy that's released from the breaking of that bond. Now that bond, being high energy, it's used up and then is used and then reforms in the mitochondria. However, if someone is, say, running very long distances, using up a lot of the third phosphate, and you end up, once you use the ATP, adenosine triphosphate, and you lose the third one, you have ADP, diphosphate, remaining. And then, so and then that can be used up for a little bit more energy, and you end up with AMP, monophosphate. Maybe I'll leave it there. The overall system, that it's just an amazingly efficient system where energy conversions occur. So, for example, light energy from the sun is converted in plants, in the chloroplast dimension, to a chemical energy called glucose. Okay, now the glucose is very, very stable and can stores a huge amount of energy through all these molecules. Now, when starch, which is what plants produce, a starch is made up of a massive, massive number of glucose molecules. That's starch. Now, ATP is a chemical energy which is used for movement. That's kinetic energy. And it warms us. So we produce, in going through the process of using kinetic energy, we produce heat energy. You can hear me through your speakers. So the speaker is using energy to make my voice heard. I am making my voice heard in sound energy. So that's also being produced from ATP. Now, when plants are eaten by animals, most of this plant material is digested and broken down to glucose. Now, starch, I said, it's a plant material. It's a huge amount of glucose. The cell walls of plants is made of glucose. It's a cellulose, and so on. Just keeps going. So cellulose, plant cells, cell walls, glucose. It's then we eat them. Now, it depends what we're eating. Some cell walls we can break down, and some we can't. So you have, for example, with bran, you can have soluble bran and insoluble bran. So the insoluble gives you the roughage, the soluble gives you the energy. So what have we got here? This is a chemical. So this is, well, the way we write the formation of metabolism. No, sorry. What do we call it? The production of ATP, C6H12O6. That's the molecule of glucose, but many other sugars as well. But it depends on how it's structured. Plus oxygen. It produces carbon dioxide and water. Okay. So this process of one molecule of sugar will produce 36 ATPs. It's very efficient. And this is called cellular respiration. This is how we live. We live constantly being alive. This process is produced. When we need more energy, we break down more sugar. And that is metabolism, if you like. Cellular respiration is the process that we get our ATP. Now, in the process of producing this, we produce what's called metabolic water, as you saw here. This water here is exactly the same as tap water, but it's called metabolic water because we produced it. So we produce it, and there are some organisms that never ever drink, or hardly ever, and they rely on their metabolic water to live. So, for example, we have two little animals, the kangaroo rat and the spinifex hopping mouse. They only eat dry seeds. Now, dry seeds are just full of starch. They do not need to drink. They live off metabolic water. And there are migratory birds that rely exclusively on metabolic water when they make non-stop flights. They fly for days and they don't stop anywhere. And they pick up the winds and they go with the winds and they fly. So metabolic water is very, very important. So what happens within the mitochondria inside the internal membrane? One glucose molecule will produce I won't go into detail, but just to show you how this happens, we'll produce a molecule called pyruvate, and in doing that, it produces two ATPs. This is outside of the molecule, mitochondria. Then this pyruvate is moved into the mitochondrion and goes through these processes, and this Krebs cycle produces another two, and the third bit will eventually produce 32. So if you add these numbers up, you'll end up with 36 ATP. So one glucose molecule will produce 36 ATP molecules. And it's a very efficient system. We have other organelles called lysosomes, and they actually carry enzymes and they help with various processes in the body, mainly in animal cells. They move, because things move around animal cells much more easily than in plant cells. These lysosomes, they actually pick out worn out cells, or the cell in areas where there are too many cells to be produced. They break them down and their material gets recycled. They may be used to destroy viruses, bacteria. If a cell is damaged, the lysosomes will help self-destruct, and this this process of programmed cell death, which occurs in cells which are a little bit getting too old or they're damaged, and apoptosis is this process where they slowly are worn down, not worn down, they break down. It doesn't take long. And the lysosomes that help with this process are just little bubbles, and they have an outer membrane which transports proteins. Now this word here, hydrolytic, so these are enzymes that help break down the proteins and other things in them. So they have these enzymes in there that contain the enzymes, and they break down excess sugar, lipid, whatever, into substances that cells can use. So everything's recycled. It's not gets thrown away. So these lysosomes help in this process of apoptosis. We also have other things called, we don't. Plants have other things called chloroplasts. This is where photosynthesis happens. Now, as we talk about energy, you get light energy, you have chemical energy, we have sound energy, we have kinetic energy where we move. So light energy is converted to chemical energy. And this is what basically what a chloroplast looks like. It has also like mitochondria, double membrane. You can see here it's got two membranes. The internal membrane holds these layers are called thylakoids, and they are linked together by this little highway of systems here, and the liquid or the space between all the thylakoids is called stroma. So different parts of photosynthesis happens in different areas. And they also have their own DNA. Now, what I didn't mention before, I was going to, and I forgot, was that if a person, any of you sitting there and say, I've got to do some exercise, and I'm going to start lifting weights, slowly build up. And as you lift weights every day or 3 times a week or whatever it might be, your muscles initially are in pain, you're sore, but that's okay because you've broken a few strands of muscle, but they get stronger when they get reformed. But the thing is that the more weight you can lift, the more energy you will need to be able to lift. So more mitochondria are produced to be able to supply your arm, your leg, whatever, your back, more ATP. So then you have this fantastic bit of life that you wanted to run or to lift the weights. And then after a few weeks, you say, I've had enough. And then you stop or you slow down. So the number of mitochondria are reduced because you don't need that much energy. It's the same with plants. If you move a plant to a higher a level of higher, or an area of higher sunlight, it will produce more chloroplasts, so it can produce more chlorophyll and produce more sugar. That's what that does. So, and it's got its own DNA, of course, like mitochondria, and so that happens. Now look at this. Photosynthesis uses carbon dioxide and water. It produces the glucose and oxygen. And this requires light and chlorophyll, which is the green pigment. And so this reaction is the reverse of the cellular one. Okay, so before we used glucose for cellular respiration, glucose and oxygen, and it went the other way. We produce carbon dioxide and water. So one's the reverse of the other. So, from glucose, I mentioned starch. We store glucose in our liver and a bit in our skeletal muscle, and the structure of starch and glycogen, which we store, animals store, is sort of similar but different. And then there's another form of glucose storage. It's called starch. Sorry, it's not starch. cellulose, and again, it's 100% glucose, but it's a different store, so a different structure. So from glucose, which is what the initial sugar is made, from that you can produce fructose, which is similar. Lactose is in milk sugar, so when the cows or animals that eat grass and produce milk, they eventually produce lactose. Of course, you can get sucrose, which is two sugars, which is fructose and glucose together, and they're produced by cane sugar. So there's a whole stack of sugars, but the first one is always glucose, and that's one it's broken down to. Now, photosynthesis only uses a couple of colors. It uses the red end and the blue end for the of light and the spectrum. It does not use the green. A little bit on the edge, but not the green, and that's why leaves are generally green. The chloroplasts in the leaves absorb these other colors for usage, but reflect the green. Now, sometimes you might see a red leaf. So there are different sorts of chlorophyll. The one I mentioned is the general one, but ones with red colored leaves absorb the other colors for usage, producing sugar, but reflect the red. Okay, so so if you see a color, I don't know if you're with me with that, but if you see a color, the dye that makes up, so for example, I've got a pen here that's sort of bluish, purplish in color. It absorbs all the colors except the ones that make up the bluish one, and that's reflected. That's what a paint is. If you've got a, I don't know, a yellow piece of paper, the yellow is what's reflected, and all the other colors are absorbed. Each chromosome now is made of DNA, and it's coiled over and over and over in histones with these special proteins, and they and these chromosomes we can code for about 100,000 different proteins. Now, the sequence of nucleotides, yeah, I'll get to those later, for each protein is in the gene. So a gene is a sequence of nucleotides which codes for the proteins which we produce. So if you've got a cell, you've got a nucleus, a chromosome basically looks like that. The banding is the old way of showing it because that's the way they used to be able to differentiate between the different chromosomes, and they're composed of DNA. The ends have got these things called telomeres, which stop the chromosomes from unraveling. So we have humans, us, we have 46 chromosomes in each cell. I think we talked about it last time. Now, chromosomes occur in pairs in each cell. One came from the mother, the maternal, and the other half from the father, the paternal. And when a new organism is produced, half the sex cells that we produce have half the number of chromosomes from each parent. They come together and we get a new individual. A gene is a bit of the chromosomes, and that codes for a particular protein. That's a repeat. That's an important statement. So we have about 22,000 proteins. We can code for these. So we have about 22,000 protein coding genes for the 100,000 proteins. And these genes are spread through the 46 chromosomes. Now, the genes that we have from our parents have been randomly selected from each parent. And using this idea that they are random, we can actually work out probabilities of what characteristics individuals may have. So with inheritance, there are a number of different basic types of inheritance. We have what's called a dominant recessive inheritance, which is what people normally understand. One characteristic is dominant to another one. We have codominant inheritance, which means that both characteristics, shown one in each chromosome, have an equal level of inheritance, of being able to be seen. Whereas with the recessive, the dominant one is dominant and suppresses that one. Here, they're both equal, so they're both seen. And then there's an intermediate inheritance where you get a sort of a balancing effect, if you like, of those two bits of information. So I've got a bit about terminology. A gene carries the nucleic code for a protein, just a protein, say eye color. The allele is a variant of that gene. Say, for simplicity's sake, let's say there are only two eye colors, brown and blue. The genotype is what genes we have. Now notice here I put the brown in the capital and a blue in little b, big B. And that's how we would say that, big B, little b. And the big B is dominant to the little b. So if you have one big B and one little b, you would see the brown color. So the genotype for two alleles is inherited. So we have three possibilities: big B, big B, big B, little b, or little b, little b. That's the genotype. It means the genetic information. The phenotype is the physical properties, what you see. So it's either brown or blue. Okay, this is just a very simple example, but it's basically the same right through. So let's have a look at these three, the dominant, recessive, codominant, intermediate. I'll explain that. So dominant recessive, selecting a single trait like eye color, we can calculate the probability of the baby's eye colors if we know the parent's eye colors. Again, simplified, another different colors, but on a simple level. So, say there are only two eye colors, blue and brown, and the variations of the gene are the alleles. We mentioned that. Now we'll say that the brown is dominant for the eye color, and blue is the recessive. So we would show the dominant with a capital letter and the recessive with a little letter. And because they're random, you can work out the probability by using what's called a Punnett square. And this is a Punnett square. So if we have a parent with a big B and a little b, and the other parent the same, by crossing, you probably remember when we were at school, we had exercise books, and always on the back, they had information of different sorts. And one of the bits of information was multiplication tables. We had a number along the horizontal, a number down the vertical, and to get a multiply 6 by 8, you find the 6 and the 8, and you multiply, then you get an answer. So we have the same here, parent 1 and parent 2, big B, little bit from each. So big B there, and then big B, little b, and with a little b here, big B, little b, little b, little b. Now, three of those because big B is dominant. 3 of those would be the genotypes for brown eyes, and so that's 3 out of 4, 3/4, and 1/4 would have blue eyes. So this is the same thing here. So we have what I just said, go through it. So we have the brown, so that's 3, the blue, that one. So we have a ratio of 3:1, which means that 1 chance in 4 of having a blue-eyed baby, or a 3 in 4 chance of having a brown-eyed baby. Now, the dominant recessive inheritance, okay, is if the 2 alleles are the same. This is still the same process. We say it's homozygous. They are the homo means the same. 2 big B's, 2 little B's. So big B is called homozygous dominant. Homozygous dominant. The big B, sorry, little b is homozygous recessive. Homozygous recessive, both the same. When they are different, this one and this one, we say they're heterozygous. Big B, little b. They're different. Hetero means different. Now, yellow peas, for example, are the dominant type of pea. The green peas are the recessive version. And with peas, for the color, they use Y's. I don't know why they use Y, but that's what I'm using because it's when it's done. So yellow peas, yellow peas are pure breeding or homozygous, okay, in cross 1, and the green pea, they're big Y, and the green peas are pure breeding. So here we have big Y, little y, and I should have gone done little y's in green, I'm sorry. And so we have big Y, little y, big Y, little y, big Y, little y, all the way through. So all of them are heterozygous, one of each, okay? The odd one, big Y, little y. That's the first cross. The second cross, you take peas from this first cross, big Y, little y, big Y, little y. So you end up with big Y, little y here. It's the same as the eyes, so the green, sorry, the brown and the blue. Big Y, little y, big Y, little y, little y, little y. So so the genotypic ratio here in homozygous yellow to heterozygous to homozygous green is 1 to 2 to 1. 1 to 2 to 1. So the phenotypic ratio, so if you come across these words anywhere, so the genotypic is the genes. The phenotypic, the phenotype is what they look like. So in this case, it's the color. So it's 1, 2, 3, which are yellow, to 1 green. So if you're looking at the inheritance of the sex of a baby, a mother has 2 X's, a father has an X and a Y. So the crossing of these, X by X, so these 2 will produce females, these 2 will produce males. Now, this system, we call them Punnett square, and again, the ratio is 2 to 2 or 1 to 1, so 50% chance of either. Now, the second version is co-dominant inheritance, not dominant recessive, but co-dominant. So this is where the genes have, or the genes have equal weight. So in human blood types, we have 3 alleles. Alleles are versions of the gene. we have type A and B for the blood types, and the D is for the rhesus factor. With respect to AB, we have AB and AB. Type O indicates a lack of an allele, no A and no B. With codominance, when both alleles show dominance, as in the case of the AB. Now, with codominance, just having a letter, you use a capital other letter. So there, for some reason, I, the letter I, capital I, was chosen, and would usually write it as on top of the corner of that, the other letter, so you have IA, IB, and that would be for an AB blood type. The others, of course, if for an A, it could be an AA or an AO. Same with the B's and so on. So we have a number of blood types. We have AB with a positive, that means RH, the rhesus factor is there. We have AB negative without the rhesus. Each of these, as you see, this A positive, B positive, O positive, has certain antibody, antigen, what's the word? Receptors. And this shows you, for example, let's start with the O negative. It has nothing on its cell membrane on the outside of it to indicate anything. It has various things, but as far as the blood types, whereas the O positive would have here the RH factor, the rhesus factor. So we'd have a few of them on the outside. The B positive would have the RH for the positive, as all these have. The B would be a particular antigen. The A is a different antigen. And the AB would have both of them. So this one is one with a lot, and this is one with just the RH. And this one has nothing, and these three are the same as those three, but without the RH. Now, this is where things can become quite interesting. My blood type is O positive, so I can donate to these three here. O negative can donate to everybody, so they're the most wanted blood type because there's no fuss, they donate to anybody. By the way, there are other blood types as well, but this is the main one. Now, if you're an AB positive, that's a really good one to have, because if you need blood, you can accept blood from everybody because you've got all these molecules on your surface. Now, by codominance in an animal, this animal, Dalmatian, would have genes for black coat, genes for white coat. Now, if there's something intermediate in between, it would be a grey dog. This happy dog, he's got splotches. So that means some parts of the skin will show the white, other parts of the skin will show the black hair. Okay, that's codominance, so equal. With cattle, you can have red cattle or white cattle, pure, big R, big R, big W, big W. And when it's splotched like this, I don't have another word, it's called roan. it's a red, but with white, called roan. Now the F1 generation, F stands for filial generation, as in I don't know what the word in Latin is. In French, it's fils, as in son. So F1 generation, roan cow and a roan bull. Get these ones, roan will produce 1 red, 2 roan, and 1 white in the second generation. So you've got these producing these roan ones, and this is what happens. The same again. So you've got big R, big R, big R, W, RW, and WW. They're all capitals, but they're equal. So in cattle, you've got that, and we saw, and the ratio is 1:2 to 1. And then we have intermediate inheritance. So you don't have, it's not dominant and it's not recessive. And you get this blending of the alleles. And so the symbol for intermediate inheritance is shown by a capital letter. Like we did here, they use capital letters, but we have a letter that's used to carry the important letter. Okay, so with intermediate inheritance, if you've got a red flower, RR, and a white flower, if you cross them, you get pink. So it's not red, it's not white, it's intermediate. So there's a flower, a Japanese flower called Japanese 4 o'clock plant. And so you've got, for some reason, this diagram also got a sort of carrying matter, big C. But so you get the red, the white, and the pink. And so there's your red. And you cross these, the CR, the RW, the CR, CW, CR, CW, you get the red, 2 pinks, and a white. So you get this 1:2 to 1 ratio again. So if you know what you're doing with plants, you can generate whatever you want. Then we get a little bit more complicated. Instead of looking at 1 gene, we can look at 2 genes. That's what it's called a dihybrid cross. And this gets more realistic. So instead of having 4 squares in the Punnett square, we use 16 of them. It comes a bit better. So if we look at a pure breeding pea plant, so we have a yellow plant, capital Y, and a round one, that's R, capital R, and they are both dominant. And the other pure breeding plant, so this is for yellow peas. Remember I said before, yellow peas are the dominant. The green peas are the recessive. So these are both recessive. So it's green and wrinkle, and they're both recessive. So I did this in color. So you had yellow and yellow and green genotype, yellow and green dominant, yellow and green. Yes, sorry, yellow and round, yellow and round, yellow and wrinkled. The gametes are big Y, big R, little y, little r, and the offspring are those 4 together. So it's yellow and round. Then you take this second generation, that one, or take the first generation to produce second generation, and where we had 4 squares, it looks complex, but it really makes a lot of sense. You end up with 16, 4 by 4. So if you take that big Y, so you have big Y, big R, oops, big Y, big R, big Y, little r, big Y, big R, big Y, little r, little y, big R, little y, little r. down one side and then down the other side here, and then you cross them. So if you cross big Y, big Y, big R with the same, you get the same. Down the bottom here, little y, little r, little y, little r. So the peas that we generally eat, we like, are these: little y, big R, something. Big R, little r, or big R, big R. So if you want only green peas, you select these, and especially this one here, and that gets rid of the wrinkles and gets rid of the yellow. And that's what we eat. For some reason, people always want to eat green, round peas. But yellow round and yellow wrinkled and green wrinkled, they all exist. But farmers can't sell them. So that's why we have all our plants are that one. All the peas that we eat are little y, big R, big R. Now, next time you have to sit around the table and explain to people why you're eating green, round peas, if you look at the eyes a bit glazing over, it'll be time to stop. Okay, now the ratios are interesting. Yellow round, that if you count them, yellow round, there are 9 of them. The green round, 3, the yellow wrinkled and green wrinkled, 3 and 1. So this is the very famous 9:3 to 3:1, 9:3:3:1 ratio for a dihybrid cross. Okay. So this is the second filial cross. So if you have this ratio and you plant 160 seeds from the first generation. All right, let's go back. That's the first generation there. So that half of them are what? They're the gametes. And they're all big Y, big R, little r. So if you plant 160 seeds of those, you will end up mathematically 90 yellow round, 30 green round, 30 yellow wrinkled, and 10 green wrinkled. So you take these 30 here, and that's what you grow. You just select what you want. Okay, eat them. Now, pea plants are self-pollinating, like tomatoes, and so you won't get unwanted crosses. So the genetics is quite simple, and the farming is quite straightforward. I like this. Why the long face? Heredity. So this is all about heredity. Now, with cells replicate, and they have 2 types of replication. One's called mitosis, and the other one's meiosis. And mitosis is when we replicate chromosomes that separate into 2 nuclei. So when you need a new skin cell, for example, the skin will produce 2 new cells, and they're absolutely identical. So you have 2 new nuclei in 2 new cells, and this type of cell division gives you 2 genetically identical cells, and the number of chromosomes is the same, identical. Now, this is the diploid number of chromosomes. Remember, almost all our cells have the diploid number of cells, half from the mother, half from the father. So all under mitosis, this is your normal replication. As we're growing and as we're shedding cells and we're renewing cells and whatever, everything's new. They all have the double number of chromosomes, the diploid. So what happens here is The normal situation of a cell is it's resting. It's called interphase. It's in between the replicating. So your normal cells around the body, they're not doing much. They're just happily being you. But if a cell needs to replicate, a few things happen. The nuclear membrane disappears. This thing here, that's called an aster, that replicates. One goes to each end. And now we're assuming there are only 4 chromosomes. And the chromosomes replicate. So you get 4 identical pairs here. This little dot in the middle, which hold them together, it's called a centromere. And it's a very important part of the chromosome. Now the middle bit, it's called metaphase. So the cell has grown, it's produced more of the internal material. The asters here throw out these very fine lines, if you like. It's not the word, but I can't remember. It's got a special word I haven't thought of for a while. And each of these, so there are 4 here and 4 there, and they get pulled towards the ends. And that process is called anaphase. And then new nuclear membrane develops, a new cell membrane develops, and the telophase is that process, and you end up with 2 new cells, identical. And here's a much more complex process, but as you see, the nuclear membrane is broken down, you have the dotted line. The cell, the chromosomes shorten and divide. The centrioles, one goes to each end, and it's more, and these names, I won't bother you about the names of them. And the same thing happened, they line up along the middle, it's called the equator, and then they get pulled to each end, one of each, like that. And the same happens, and you end up with 2 brand new cells. That's called mitosis. Spindle fibers, things. Thank you. I've forgotten. So they're called spindle fibers. Now, I think I'm getting old a bit. The other type of cell replication is called meiosis. And this is where we produce the replication. The sperm, we produce the eggs, and they have half the number of chromosomes. So instead of having 2 of every chromosome, they have a single set. So they don't have the diploid, they have the haploid. So one cell that's undergoing meiosis produces 4 gametes, whereas here it produces 2 gametes with all our chromosomes in each. We produce 4 cells, 4 gamete cells, 4 sperm, 4 eggs, and they have half the number of chromosomes, the haploid number. So in males, each meiotic cycle, the meiosis produces 4 sperm, and in females, they do produce 4 cells, but one is a huge one, the egg cell, and the other, what's left, are 3 tiny cells which eventually just get resorbed into the body. So during the meiotic cycle, they separate, the chromosomes separate randomly into different gametes. So that means you get a mixture of parents, right? The original parent chromosomes are mixed around, so no 2 gametes are produced exactly the same. So during the meiotic cycle, the cells divide twice, whereas during the mitotic cycle, the first one we looked at, they only divide once. This is for the production of sperm and eggs. Now, the process here is different to mitosis, where in mitosis, they line up on the equator and homologous, the same, homologous, the same chromosomes are next to each other. And this is in meiosis. So they line up the diagrams in a minute. So they line up next to each other. So then a process called crossing over occurs, where they cross over and swap information. Okay, so here we have the original father's paternal chromosome. Now they've replicated. Okay, so there's two of them. The original mothers. Now they move around, and then they touch. Where they touch, they break apart and reform. So these are, I'll get some words here. Homologous means the same. Let's say chromosome number 1. So they're both number 1. It could be chromosome 16. They're both the same. This is called the crossing over. So chromosome crossover. And these are called recombinants, because they have recombined material from each other. Now, even though these are originally these are one chromosome, which is divided into two, because they're still joined together, they are called chromatids. And they're joined at the chromosome, as at the centromere, sorry, at the centromere. So each of these is one chromatid. And these ones with additional bits, this one here, they're called, these are called the recombinant chromatids. And these outer ones with the haven't joined are non-recombinants. Okay, so instead of having two the same, now we've got 4 different ones. Two there. We've got 2, now we've got 4. So that the number of recombinants varies each time. And where the crossing over occurs varies each time. So you get your children have some similarities to you and your wife or husband, your spouse, I should say. Your great-grandchildren have similarities not only to their parents, but also to their grandparents because of this situation. And this is the same thing going on. So I won't go through it all. But I can't remember whether I put it into the notes or not. But there are 2 phases. You get the first phase, you get 2 cells. And then in the second phase, you get the same going through, but you get 4 cells with half the number of chromosomes. And each of these in males produces a sperm. In the female, one of these is a very large one, and you get 3 little ones. Okay, here we are. So 2n is the full number of chromosomes in a cell. Remember that 2 of each chromosome, so called 2n. We get through the process of meiosis, and you get half of the number of chromosomes, not 2n, but n, and you end up with 4 sperm with this half the number of chromosomes. In the female, the same thing happens. You get the 2 ends dividing down at this level, but from here you get you end up with 3 of these tiny little ones, which disappear out the world, and you get 1 large one. Well, 1 large one there. This one shows fertilization, the sperm coming in. Oh, here's sperm coming in here. Completion of meiosis. Okay, that's it. I won't go there. Now, how about some numbers? We have about 100 million sperm produced daily when we're most fertile. I don't know when males stop producing sperm, or if they ever actually stop, but the numbers reduce as we get older. But men in their 50s, 60s have produced babies in the past. I don't know how good the sperm are. I don't know how many there are, but obviously there are still some there. For us to produce sperm, it takes about 3 months. And I went through that last time, 2 weeks ago. It takes about 3 months from production to maturity. Now, spermatogenesis, that's production of sperm. We produce many millions of sperm a day. It's estimated we produce about 1,500 in our heyday per second. And no wonder we guys feel tired all the time. With females at birth, they're about between 1 and 2 million eggs. By the time of puberty, there are about 300,000 remaining. Of these, about 500 will be ovulated. Now, if you remember back to the last lesson, to the DNA structure, we had 4 nucleotides: A, adenine, thymine, cytosine, guanine. So I like this for the teacher. Okay, class, let's say our alphabets, A, T, C, and G. This is just a reminder for you. Now, DNA is always single. DNA is always double stranded. RNA is, we used to say, always single stranded RNA. We now found this a couple of viruses with double RNA. So this isn't quite right, but I like it. And guys, here's where we stop. I hope you're still there.
Yep.
Yeah, there you are. I see.
Hi, it's Michael. I just want to say thank you to everybody. It's been wonderful. This is going to be my last one because I'm heading back. We're heading back to the States at the end of the month. And if I did join you, it would be 3 in the morning. I don't think you'd want that.
But you can always catch up with it on YouTube.
Yes.
If you want to. You know,
of course I'll do that. There was, you know, when I would have my residents, I would always ask them why we breathed oxygen. And you really pointed it out beautifully because of the differential between 2 ATP and 36. That if you went down if the electrons went down the mitochondria,
yeah.
The last molecule that would accept electrons was oxygen, and that's why I think that's why we breathe. That's why oxygen is so efficient in the mitochondria. And the other thing that I always found interesting was thinking of this notion that we truly are children of the sun, that with nuclear fusion, you eventually get electromagnetic waves, which come all the way to the earth, that's converted into sugars by plants. And you know, we're from that. So if it weren't for the sun,
we wouldn't exist.
Which is something to think about, because apparently we have it's already halfway done. So there are only 2 billion more years left. That without the sun, we're going to be in trouble.
Yeah, I'll have to think about where will I be in 20 million years, billion years rather, or 2 billion years, whatever.
All right. Anyway, thank you so much.
It's something to think about for our generations, but with people like Putin around, I don't know how much longer we'll be here for. So,
Well, I suspect that Putin won't be. I suspect we will outlive Putin.
I'm sure we will. I'm sure we will. Anyway, Michael, it's been lovely having you aboard for a short period.
Thank you.
And if you're ever in Melbourne again, you know where we are.
Yep. Okay.
Okay, guys. Some of you heard me talk about this earlier. Just before you go, on Monday, Pat and I are going to Adelaide to support her sister, whose husband is very, very ill. So I'll be missing the next one in the fortnight. A fortnight after that is Cup Day. And I think we've got something planned. I don't know. I didn't do the planning. And fortnight after that will be the last one for the year. So it's, you know, I love doing this. I've been retired from teaching for 15 years now, and I still get a buzz of.
No, you only think you've been retired. You haven't really.
You get well paid, Ruben.
I know I do. Yes, you should see my bank account.
Yes.
You know what? Listen, those of you who are Jewish, Chag Sameach, and fast well if you're fasting, and if you're not 100% healthy, make sure you eat something in the morning, or at least or drink some sugar. Sugar is important, and we'll just see what happens. Just take care, and I'll see you in what's that? About 6 weeks for our last one.
Yeah.
All the best, Ruben.
All the very best.
Thank you very
Michael,
much.
safe travels.
Thank you so much. Thank you, everybody.
See you.
Bye bye. Bye bye.